Assalamu alaikum. My name is Mahanur Khan. Roll number is EP1770084. My topic is hypoglycemic effect and subtopic is diabetic agent glucose transporter 2. Glucose transporters. Glucose transporters are a wide group of membrane proteins that facilitate the transport of glucose across the plasma membrane, a process known as facilitated diffusion. Why we need glucose transporters? As we know that glucose has many hydroxyl groups that make it hydrophilic in nature. So by glucose transporters, it is easy to transport them across cell membrane. There are two types of transporters, glucose transporters and sodium dependent glucose transporters. We have 14 different types of glucose transporters. They are sodium and ATP independent. They are present all over our body. Whereas sodium dependent glucose transporters, they are sodium dependent, require ATP and located in intestine, renal tubules and the blood brain barrier. GLUT1, they are present in blood, blood-brain barrier, heart, but in lesser extent. They are insulin independent, that is, they never need insulin to uptake glucose. GLUT2, they are present in liver, pancreas, and small intestine. They are also insulin independent. They have low affinity for glucose, that is, they take glucose when the glucose concentration is very high, and uptake glucose as glycogen. Whereas GLUT3, they are present in brain, neurons, and sperm. They are also insulin independent, but they have high affinity for glucose. Whenever there is a glucose, they uptake them. GLUT4, they are present in skeletal muscle, adipose tissue, and heart. The important thing is that they are insulin dependent. GLUT5, enterocyte of intestinal epithelium, they are also insulin independent. They uptake fructose. The function of rest of new glucose transporter isoform is still not clearly defined. Glucose transporter 2. Glucose transporter 2 is a transmembrane carrier protein that enables protein facilitated glucose movement across cell membrane. It is the principal transporter of transfer of glucose between liver and blood and for renal glucose reabsorption. Glucose transporter 2 is increased by hyperglycemia in liver of diabetic rat. Now, there is an article that have evidence that glucose transporter 2 increased by hyperglycemia in liver of diabetic rat. Now, in this article, we performed the experiment. In this experiment, animal male Wister rats, 250-gram breed in our laboratory, were housed at 240 degrees centigrade. They had free access to water and food. These studies were performed on conscious rat 24 hours after implantation of catheters into the two jugular veins under phenobarbital anesthesia. Immediately after surgery, rats were randomly distributed into four different groups and housed in individual cages. Group number 1. This group has rat had free access to food and water. They are killed. This group is termed as fat controlled rats. Group 2. This group of rat was injected with streptozotocin through the left jugular vein at a dose of 65 mg per kg body weight. Rats had free access to food and water and were killed 16, 6, 18, 30, 48 or 72 hours after the streptozotocin injection. This group was termed as diabetic rats. Group 3, a group of diabetic rats was infused intravenously with fluorizin at variable rate after streptozotocin injection to 72 hours to maintain their blood glucose level in the normal range. They had free access to food and water during the experimental period. This group was termed as fluorizin treated diabetic rats. Group 4, a group of streptozotocin diabetic rats was infused intravenously and insulin at a constant rate from 18 hours after streptozotocin injection to 48 hours to maintain their blood glucose level in the normal range. They had free access to food and water during the experimental period. This group was termed insulin-treated diabetic rats. In some of these insulin-treated diabetic rats, insulin infusion was stopped after 48 hours. The rats were killed 6 or 30 hours after insulin withdrawal. Then, after that, we monitor the blood glucose and plasma insulin and glycogen level. After that, killing of these animals occur and tissue sampling. Then total RNA were isolated from liver, then quantification of glucose transporter 2 by immunoblotting occur. After this experiment, the result that we have is glucose transporter 2 liver of fed non-diabetic rats were used as control. These values were considered as 100%. Glucose transporter 2 concentration decreased by 90% as soon as 6 hours after streptozotocin injection and increase after 30 hours to reach level two-folds higher than in non-diabetic rats after 48 hours and later. Normalization of blood glucose concentration by fluorescein treatment of diabetic rat prevent the increase in glucose transporter 2 protein concentration. In insulin treated diabetic rat, when insulin treatment was stopped, an increased glucose transporter 2 protein concentration was observed after 30 hours. So, through this experiment, we concluded that glucose transporters to increase by hyperglycemia in liver in diabetic rats. There is a reference. Thank you. Allah Hafiz.